Thank you very much. Well, thanks for asking me to come and talk about this, and I'll try not to deepen your postprandial slumps too much. So as Volker said, yeah, I'm now based at Genomics PLC in Oxford with an honorary appointment at UCL, and Genomics PLC does exactly what Maureen and Volker were discussing at the beginning about integrating genetics throughout the drug development process. And I'll try and illustrate a bit about how that works. So I think you're very familiar with this problem that developing new drugs takes a long time, costs a huge amount of money, and often goes wrong at the very last minute. This problem of late-stage attrition, and there's a growing argument that genetics can be used to try and reduce the rate of late-stage attrition by trying to get more information about the validity and safety of a drug target earlier on in the drug development process without having to wait for the phase 3 randomized trial to make those decisions, which often come far too late, and by that time many patients have been put at risk of a potentially ineffective or worse, a harmful new medicine. So I'm going to talk a bit about what are the properties of genetics that allow us to do that, and then show you some examples of how it's been used and how it might be used. So the title of the talk is Nature's Randomized Trials, and it's a little bit of a leap, this analogy, but I think it's quite a helpful one. So we're all very familiar with the design of the classical randomized controlled trial, where patients are randomized to the intervention arm or the control arm. They're followed up over a time period. Changes in their health, their behavior, their biology are measured over that time, and any difference that's observed between the intervention and the control arm is ascribed to the effect of the intervention, because by virtue of the fact that they were randomized in the first place. So by randomizing those patients, you avoid the effects of potential confounders of your observed effects. That's the basis for everything I'm going to say from now on. In nature, we have a very similar sort of scenario, but without having to do anything, without having to give anyone any medicines, because at the time of your conception, you're randomly allocated to either an intervention arm or a control arm for any given point in your genome. So the two arms of your trial are the two alleles for any given point in the genome. And we can look at people who have one allele and compare them with the people who have the other allele at that point in the genome, and compare all sorts of things between those two groups, whether it's a biomarker going up or down, behaviors being different, risks of a disease outcome being different between those two groups. And because the allocation to your genotype is random, it's a 50-50 chance which allele you receive from either your father or your mother, that randomization process and its consequences for inference from what you observe in a genetic study is the same as what you'd have in a randomized control trial. So we can, if we observe a difference in a biomarker between the group with one allele compared with the group with the other allele, we can infer that difference as a consequence of that genetic difference, that genetic variation. So a couple of other sort of ancillary benefits of Mendelian randomization, apart from this intervention, which is your genotype and the randomization process, is that by definition, these experiments, these Mendelian randomization experiments, are longitudinal. They start at the time of conception, and follow-up continues until you die. So we therefore have this really nice, very long-term follow-up period, and we'll see how that's important and how that may be something to consider in designing these experiments later on. And these are, in general, blinded experiments as well. For the vast majority of your genome, you've no idea what allele you hold for any given variant, except perhaps if you have ginger hair or it's something that determines your skin color. We're almost entirely blinded to what our genotype is. So we have that benefit as well of avoiding bias through changing behavior or otherwise through knowledge of our genotype. Another chief benefit of this type of experiment is this avoidance of reverse causation. So we know that there's this one-way street of biological information, starting from your genome sequence, translation into messenger RNA, and then further down into protein, more complex traits like blood glucose or BMI, and then into disease, coronary heart disease, stroke, diabetes. So there's this one-way flow of information. So if you perturb biology here by selecting a genetic variant in a gene that you're interested in, everything that you observe in addition to that should, in theory, excuse me, be a downstream consequence. So the direction of the path of causation goes that way, and with a very few exceptions, can't go that way. So we can make further inferences on that basis. So that's the genetics bit of this. And Mendelian randomization is often displayed in a graph like this, which is slightly complex looking. So let's apply it. So you want to ask the question, does higher CRP, high circulating CRP, cause coronary heart disease? Fair question. It's been addressed widely and controversially, but let's stick with it for now. You could measure CRP in people with and without coronary heart disease, and you'd see that the people with coronary heart disease had higher CRP than people without, and you could make the inference, therefore, even if, whether you do a cross-sectional study or a longitudinal study, you could make the inference that CRP causes coronary heart disease. But we know that this is a hugely confounded relationship. There are a number of things that influence both CRP levels and coronary heart disease risk. So that on its own doesn't give you the answer about whether CRP causes heart disease. We can then bring in genetics. So if you take common variants in the gene that encodes CRP, C-reactive protein, those genetic variants influence CRP concentration, but shouldn't influence potential confounders. They may influence things that happen in a biological pathway between CRP and coronary disease, but not these side pathways that might confound this relationship. So you've got your genetic variants that affect CRP levels. You then want to test whether those same genetic variants associate with risk of coronary heart disease. If they associate both with CRP levels and higher coronary heart disease risk, then you're in a position to say, yes, CRP causes coronary heart disease. In actual fact, the opposite is true, and they suggest that there's no causal relationship between CRP and heart disease. But you can substitute in here, instead of CRP, body mass index, blood glucose, interleukin-1, we'll come back to that, blood pressure, more or less anything we can now start to develop genetic instruments for that we can ask these sorts of causal questions, and avoiding these problems of confounding and reverse causation and bias that we talked about just now. So the big question is what's the relevance of this to drug development, drug target validation? We're in a fairly fortunate position in that the vast majority of drug targets are proteins and therefore have an encoding gene. We know now quite a lot through big sequencing projects, big genotyping projects, about the variants in the genome that influence concentration or function or morphology of these proteins. We also know from some really insightful work from Matt Nelson and others at GSK a couple of years ago that when you look at known successful drugs and their targets, if you have a target in a gene and that gene has variants that are associated with a disease at a strong enough level, you're far more likely to have a successful drug for that target. So genetic validation of drug targets empirically improves your likelihood of developing a successful medicine. Now, there are all sorts of caveats and biases in that, but the data suggests there's at least some benefit. So this is something we should be pursuing, and this is why we're here. I recommend this paper in Nature Genetics a couple of years ago. It's a really excellent read. So let's start with an example that's been done almost to death, but it's a very useful example. So statins are the medicine we probably know most about in the world. So we want to use Mendelian randomization to try and validate HMG-CoA reductase, the target of statins, as a target for lipid lowering and for reduction of cardiovascular disease risk. So we take a common variant in the HMGCR gene that encodes this protein that's the target of statin drugs, and we show that for each additional copy of the G allele that you have at this particular variant, excuse me, you have lower total LDL and non-HDL cholesterol in this quite nice dose-response sort of a way. So this is similar to what you might see in an early phase drug trial, in pattern at least. So we have a variant here that mimics quite nicely the effect of statins on LDL cholesterol. What effect does it have on risk of coronary heart disease? So this is work from Brian Ferentz, again, a couple of years ago. And the variants, so this uses a group of variants in HMGCR together, but the inference is the same. So this group of variants reduces LDL cholesterol in a similar dose-response way to what we just saw. and also reduces risk of coronary heart disease. So it seems to work, at least in pattern and direction. One important thing that we'll come back to towards the end is about the magnitude of the effect. So these common genetic variants, as Volkert said at the beginning, tend to have very small effects on phenotype, at least the ones that we've looked at so far. So this graph, each of the coloured dots is a different statin, the different colours, the different statins, and the little numbers which you can't see are different doses of each statin, going down right to the bottom to 5 milligrams of fluvastatin. The genetic variant that I showed you with the effects on lipids has an even tinier effect. So each copy of that additional G allele reduces LDL cholesterol by 0.06 millimoles per litre. So these effects are really tiny when you compare them to the effects you deal with in trials. So it's something to bear in mind, and we'll come back to that. So we've dealt with intended effects on biomarkers and disease outcomes, but what about adverse effects? Can we use genetics to investigate adverse effects? The answer is yes, but with the important caveat that you can only investigate target-mediated adverse effects. So if you're using genetic variants in the target encoding gene, by definition, you can only look at consequences of perturbation of that target protein. So if we take the example of statins and diabetes risk, so new onset diabetes risk with people treated with statins, we know this is a well-established adverse effect of statins, and we use that same genetic variant that influences lipids, coronary disease. We see that it has effect in the opposite direction, going up on body weight, BMI, on anthropometric measures, so waist and hip circumferences. And when you look at the associations of that same variant on diabetes risk, we see that goes up as well. So this genetic variant is modelling both the intended on-target effects on lipids and cardiovascular disease, but also unintended on-target effects, safety signals on diabetes, BMI, also other glycemic traits, glucose, insulin, HbA1c. So the validation is both for intended therapeutic effects and also for safety signals. And bringing ourselves up to the 21st century, a similar pattern is true with PCSK9 inhibitors. And when you look at genetic variants in PCSK9, the gene that encodes PCSK9, you see that there is a reduction in the odds ratio, or the odds for myocardial infarction, and also an increase, interestingly, in the odds ratio for type 2 diabetes. So there's an interesting potential safety signal there that I think everyone is keeping a very close eye on. And that was demonstrated by Brian Ferrance last year and by our group at UCL earlier on this year. So that's the proof of concept. What about repurposing and indication expansion? This is a big area of interest where you've got an existing asset, either with an existing indication or an asset that has been through some early development and doesn't have a licensed indication yet, and you're thinking, what else can we use this for? So I'll walk you through an example that uses the interleukin-6 receptor. So we thankfully have quite a well-described genetic variant in the gene that encodes the IL-6 receptor. And we know the IL-6 receptor is the target of a couple of drugs, notably among them is tocilizumab, which was developed as a monoclonal antibody developed for treatment of initially severe juvenile rheumatoid arthritis and has since been used for a number of other autoimmune diseases very successfully. We also know that higher interleukin-6 levels associate with higher risk of coronary heart disease. So the question is, does interleukin-6 signaling play a role in the development of coronary heart disease, and could it therefore be a target for intervention or prevention of coronary disease? So we have this well-described functional variant that changes how the interleukin-6 receptor behaves. And when you take that variant and look at its associations with a whole bunch of circulating biomarkers and compare those associations with the effects of tocilizumab, so this trial for this drug for rheumatoid, on those same biomarkers, and the tocilizumab effects are from randomised settings, so they're robust enough to make this comparison, we see this very close directional concordance in the effects of the genetic variant and tocilizumab. So in a similar way to what we saw with the HMGCR variant mirroring the effects of statins on lipids, we've got a similar sort of pattern of effects of tocilizumab and the effects of this variant in IL-6R. So given that directional concordance, can we therefore use this same functional variant to look at the impact of IL-6 receptor signaling on coronary disease risk? And the answer is yes, and we demonstrated that that same variant that mirrors tocilizumab action is associated with lower risk of coronary heart disease, suggesting that this pathway at least has some causal role in the development of coronary heart disease. Now, whether tocilizumab is the right mode of intervention for this in perhaps a primary or secondary prevention setting is up for grabs, but at least the target appears to be getting towards validated for use in an indication completely different to that for which the drug was originally developed. And now in the era of thousands of genome-wide associations of these large biobank data sets, searching for alternative indications or expanded indications is relatively straightforward. Now, bring us right bang up to date, could we have foreseen the findings of CANTOS? Interesting question. So this has been addressed by Daniel Freitag and colleagues when he was at Cambridge, and Daniel's closely involved in this project. And I'll show you his data in a moment. But IL-6 signaling is not straightforward. Interleukins seem never to be straightforward. So canakinumab, the drug used in CANTOS, is a monoclonal that binds IL-1 beta, and IL-1 beta binds the IL-1 receptor and has its downstream effects. This distinction becomes quite important when we look at the genetic data. Another drug, anakinra, is a recombinant form of the IL-1 receptor antagonist. So this is naturally occurring circulating antagonist of IL-1 receptor signaling. And when you give patients anakinra, it blocks the receptor to both IL-1 beta and IL-1 alpha, whereas canakinumab is specific for IL-1 beta. So when you use genetic variants in the gene that encodes that circulating receptor, that remember blocks both alpha and beta signaling, you see that it has a strong effect on these blue dots represent the effect of the genotype on IL-1 receptor antagonist levels. So it has a good, strong effect on the biomarker, but it also seems to have a strong association with increased risk of coronary heart disease, myocardial infarction. So this seems to be at odds with the findings of CANTOS. But importantly, the biology of those two experiments differs quite considerably because of these two different ligands for the receptor. So the genetic data I just showed you model total blockade of this receptor, whereas CANTOS looks at blockade just of IL-1 beta signaling. So these experiments can be done fairly straightforwardly, but they require some knowledge and some understanding of the biology, otherwise you can be totally misled. If we didn't know what we know about IL-1 receptor signaling in its totality, that genetic evidence could potentially have derailed CANTOS, and we wouldn't have seen these effects, or at least called the trial into question before it finished. So these experiments are really easy to do. I think Bram is going to walk you through some examples later on, but doing them without some really careful thought and design is dangerous. So I've shown you some examples. Like Spiros, I want to just show the other side of the coin and show some challenges that are encountered in designing these Mendelian randomization experiments. And the first is about these small studies, the small effect sizes. We saw before that this variant in HMGCR has a very, very tiny effect on LDL cholesterol. And the plot that I showed you from the IL-6 receptor tocilizumab study, you'll notice that these axes, the scales on these axes are on a couple of orders of magnitude different, because the genetic effects are far, far smaller than the effects of this potent monoclonal antibody. And as a corollary of that, the effect sizes are tiny of the genetic variants, But you have to bear in mind that those effects have been exerted right from the time of conception all the way to the point at which you make your observations. So there's this lifelong influence of the genetic variants compared with the relatively brief effects of a drug trial, which may last 1, 2, 3, 4 years on average. So even though you're using a very potent modulator of your target, it's only for a very brief time and usually quite late in life, whereas your genetic variants have been exerting their effects right from the beginning of life, albeit in a small way, but throughout that person's life course and development. As a function of both of those observations, very, very large samples are needed, and this is where these very large consortia of multiple studies come together to do these analyses. So the data I showed you from the PCSK9 and diabetes analysis had over 500,000 patients in it. The HMGCR and diabetes analysis is just over 200,000. Now with the advent of UK Biobank and other similar very large data sets, these are becoming increasingly doable and increasingly reliable. But they do require quite a lot of coordination of different studies. On a slightly deeper level, as we saw with the IL-1 receptor example, the biology may not be clear or may require further investigation to understand what your genetic findings are. Sometimes that's possible, sometimes it's not possible and requires further experiments to find out what's going on, whether it's in humans, model organisms, in vitro tissue. This question about druggable versus undruggable is interesting and hotly debated. But it might be that the protein that you're interested in as your target proves, for whatever reason, not to be druggable. But that's not a reason to abandon your Mendelian randomization analyses. What you might see is there is a protein upstream or downstream of the protein that you're originally interested in that you can instrument genetically, and that might be a potentially druggable target. So these sorts of analyses can be used to kind of shuffle up and downstream in causal pathways to see what might be there. The data substrate for doing these studies is growing. This is just a cartoon illustration of all of the chromosomes, and each dot is a genome-wide association study for one trait or another for each of the marked loci in the genome. And this is an old version that's even bigger now. We've got thousands of GWAS studies for thousands of traits, increasing now that we have very large-scale proteomic platforms like SomaLogic and other large metabolomic platforms, imaging data, EHR data. It'll expand, and the capacity to develop genetic instruments for an exposure or a target that you're interested in is and will continue to grow. And also the capacity to test that target in biomarkers or outcomes that you're interested in will also continue to grow. So I think that's more or less done. I hope I've shown you at least snippets of how we can integrate genetics into all sorts of parts of the drug development process. We've dealt mostly with early stage development, but there are opportunities for building genetics into trial design, into investigating adverse effects post-marketing. So genetics has a role throughout that drug development pipeline and can feed back and forth. And I think that's everything. Thank you very much.
I think the question about the upstreams and downstreams of if a target is not druggable, how you proceed from this knowledge to what would be druggable? Would you then this requires quite some knowledge, right? What is the way to find a druggable if the actual one is not druggable?
So I guess part of that is a medicinal chemistry answer, which I'm not at all equipped to provide. But if we take the IL-1 diagram, say it proved impossible to make a monoclonal that blocks IL-1 beta, but we knew that IL-1 beta played some role here. Maybe there's a way of binding the IL-1 receptor but maybe in a selective way that only influences IL-1 beta signaling. So you can use variants in the IL-1 receptor gene, or its associated protein, or the IL-1 receptor antagonist. So you have to have a bit of knowledge about the biology, or use biologically agnostic ways and try and infer up and downstream biology in silico, I guess, through machine learning methods. So it helps if you've got some idea of the biological environment that your target is working in, but there are probably other ways of inferring up and downstream as well.